which has lost its function in, we will see that in the next lectures, lost its function and become actually causes cells which have the mutations to become cancer. And this is one of the examples to follow this journey, where we have first, second, third, and fourth mutation will be a cancer which Dr. Aziz's lab studies. It's called colorectal cancer, and we know very well defined stages where cancers can actually show different abilities to attract and progress towards their metastasis journey. So starting with the polyp, and might take Dr. Aziz 20 years for cancer to metastasize to viscera and so on, and each of them may be accompanied with different set of mutations. In some examples we know them, in many other examples we don't know them. So if I ask you this question, DNA repair, is it good or bad? You can think about this in your own terms. Is DNA repair good? If you're a normal human being, yes, 100% we need good DNA repair system. There are people who have on an average have lives in the world over 100 years, talking about some parts of North India, extreme up north, Ladakh and so on, and North Pakistan. People live as a norm 100 years plus, may have very good genes, and when I say good genes, maybe a very efficient DNA repair system, coupled with, of course, good air quality, smoking seen as a bad thing, as bad societal trait, and they might have a very good chance to live. And of course, DNA repair is good, but DNA repair is bad for cancer cells, and we will see this in the subsequent lectures. So, Dr. Aziz, are you?
Well, I can tell you one positive aspect
Sure.
about loss of DNA repair.
Yes, please.
When using the radiosensitizers like iododeoxyuridine, this
Yeah,
is a radiosensitizer which is helpful for radiation therapy.
that's
So
correct.
people who are mismatch deficient, they respond better to these therapies and these radiosensitizers. So why? Because their mismatch repair status is lost, and they
That's
can
right.
retain more of this, and then they get better response. Second part, in the immunotherapy against checkpoint inhibitors, it's well known that if you have got mismatch repair deficiency, you will be able to generate better epitopes for
That's right.
the success of the immunotherapy. Otherwise, you don't even respond. So now it's a standard that somebody who is DNA repair deficient will be given the checkpoint inhibitors, and then those who are proficient will not be given
That's
these
right.
check.
Microsatellite instability.
Yeah.
That's absolutely right. Yeah, absolutely. These are all great points. Certainly makes a lot of sense. So we are already heading, as Dr. Aziz was saying yesterday, towards personalized treatment based on the tumor characteristics and how good we are to differentiate between these tumor qualities from one individual to other at a population scale. So, Dr. Aziz, would you like me to carry on with the rest of the talk and then the tutorial?
Yes, I think 4 to 5 was your tutorial schedule. So we can take a short break if you want.
Okay, yeah. Let's meet in.
So we have been speaking. And meanwhile, the participants can put their questions in. We'll take a short break and then we'll resume.
10 minutes, Dr. Aziz, is fine.
Yeah.
I'll leave the slide.
So I have posted the link for all the lectures, the Google Drive. So the participants can look at this and they can find my lectures there. I've already uploaded. And I would request Swati to please upload today's videos as well in my folder. And I hope Dr. Asim will be putting his lectures in the folder as well.
I will do that. Thank you.
I will have real-time access to all these materials that we are talking
I'm
about.
always running behind, so I have this lecture to cover before the tutorial, and that's why I leave it until the last minute, because I never finish my lectures on time. I get so taken by my lectures.
Totally understand. And let's, there are about 59 participants, but please make this session vibrant, asking questions, discussing. This is what we need.
it should be interactive, really, both ways, because what I do tell my students is, use this as an opportunity to, you know, get rid of your, if you're very shy, if you have any inhibitions to talk in public, and use this as an opportunity to prepare yourself for any upcoming interviews, for example. That would certainly help.
Yes, because see, there's no question of that question. So just ask.
That's right.
It's not like I don't know it, everybody else knows. No, come out of that inhibition and ask. Even if you are asking why DNA is having AGTC or AGUC, it's fine. Not a bad question.
100%.
So once again, I would encourage and request and exhort you all to ask as many questions as you can. Give us a hard time.
Please do. I hope everyone is back and we can start.
Yes, there's a slight drop in the number, so it was yeah, 10, 15 people. They just went on a break, longer one.
Okay,
But
we are
yes, we
recording
can,
this anyway, right?
we can start. Yes.
Okay, so yeah, let's just start where we stopped. Good. So I'm not going to go into the details, and this is relevant to the repair systems, and that's the reason I'm showing you this slide. What you can appreciate is that the 5' end of a nucleotide chain is always a phosphate you will find, and this is connected with a phosphoester on both sides, meaning it's a phosphodiester bond uh, through the another nucleotide. The 3' hydroxyl is where the new nucleotide, incoming nucleotide, comes. I believe you all know this. What you also know here is that this bond is called the glycosidic bond that connects the base, the nitrogenous base, with the pentose sugar. Loud and clear, I'm sure everyone knows this. And then there is this hydroxyl at the 3' end, which acts as a substrate for the growing polynucleotide chain. I'm sorry. Uh, and there are repair systems, of course, that are needed in the body to correct these defects that happen to the DNA. Uh, what I was telling you about the damage to the DNA, there were three prominent ones. So the oxidative stress-induced oxidative damages. There was second one, which was the hydrolysis of nitrogenous bases, specifically purine hydrolysis, and the third one was the deamination. So we have discussed these three. All of this, you would see that into the repair process, they will lead to generation of the single strand breaks. Do we have to worry about single strand breaks? Not really, because we have efficient systems to repair single strand breaks as well as double strand breaks. The only difference comes about is that single strand breaks are very well tolerated by the cell. Plenty of single strand breaks are generated in a cell all the time. Uh, what is not tolerated by a cell, meaning a cell will not be able to continue its normal life cycle, is if they have a double strand break. Would you believe that as little as 3, 4 double strand breaks are sufficient to kill a cell? If it is not repaired, the cell will die. And that is in terms of normal cells. If the cells are not really cancer cells, they cannot sustain life if they have any unrepaired double strand breaks. And the way these radiotherapeutic treatments, anti-cancer drugs, cytotoxic drugs function is by destroying the DNA. We have pathways which can correct these double strand breaks. Uh, it, in particular, this homologous recombination pathway, which we will try and cover in the tutorial if we get to start and finish the tutorial today. If not, no big deal. We can catch it up tomorrow at the same point. But the whole essence of this is to cover things which you will find very hard to get in any books or research articles. Um, and that's why I'm taking my time to allow you to understand this. If some of you understand and not everyone, I'll be more than happy, of course, if you understand all of it. Students I'm talking to, but yes, uh, please let me know if there is something which is not sure in your mind. So what happens here, these are some enzymes that act on a double strand break. For example, you see these enzymes which will cut the phosphodiester bond and leave a nick in the DNA as shown by this scissors. And this is caused by an enzyme called endonuclease. There are enzymes working to oppose this function, and they can seal this nick back. And these are very well known as ligases. I'm not going to go into the details. You can read that very easily in any books of molecular biology. And these are some textbook enzymes. The only intent I have is to show you how these things actually work when it comes to the DNA repair, especially in context of cancer. You have enzymes which start cutting not from the inside but from the edges of the DNA. We call them exonucleases enzymes, and these three are very important in the integrity of the DNA, especially these ligases, to make sure any nicks are any created during the repair process, because eventually you will create these nicks as you start to repair the DNA. And then these enzymes are there to take care of any defects. So the first part is this enzyme, DNA polymerase. You can read all of this information if you like in your own time. I'm going to focus on the crux of this slide in the interest of time, but also, as I said, this is something of very relevance to this talk. So what happens is DNA is synthesized in 5' to 3' direction. We all know that 5' phosphate to 3'. And this is the ability of the enzyme to carry out this function, this polymerase. But they also have a different function in opposite direction, meaning 3', not from 5' to 3'. They act as a polymerase, but in the other direction, they act as exonuclease, you can say. And meaning here, what is that they can remove any nucleotide which is wrongly incorporated. So if you see this as a template DNA, this is the new strand being formed. And what you see here, the enzyme, unfortunately, glues a wrong nucleotide. There should have been a T. Is there a T here? No, there is a C. It's become part of the DNA. It is removed under normal circumstances. So if you have these enzymes, most of them, almost 99% of them are proteins. We know that very well. What happens to the protein coding genes? If those genes are mutated, these enzymes are going to lose their ability to correct these, identify and correct these situations where a new wrong nucleotide has been put. So this function is called proofreading function, and sometimes the DNA polymerase will not have this proofreading function thanks to the mutations in the very enzyme, which was meant to repair that DNA, and that will leave some mismatches behind. You can see what happens when you have these mismatches. These tumors will have, because these DNA polymerase is now mutated, they will have super high mutation rates compared to normal cells. So that is one of the initial things happening. Cells which are not yet abnormally dividing, will be very hard for us to identify those cells. There may be just few cells in a big organ which have started to have very high mutation rate, thanks to the mutations in enzymatic activities such as this enzyme, just proofreading activity of this enzyme. And because of this, they will have sometimes new proteins being formed. Why? Because if you have mutations, these may be mismatch repairs. Sometimes what will happen, if they are not repaired, they will give rise to proteins where some amino acids may be different. And as a result of this, the codons will change, amino acid will change, some amino acids. And as a result of this, the protein will change as well. How many proteins? Well, a lot of them. So they will have a hypermutated phenotype, these cells. They will make new antigens, meaning with regards to the immune system. As Dr. Aziz was telling, these are the cells which will be an excellent candidate for immunotherapy if they were to become cancer. So first question first, how our body repairs them. There is a pathway, and you can see any good textbook that will talk about these mismatch repair pathway. This is called mismatch excision repair, which needs these enzymatic pairs, these proteins, MSH2 and MLH2 and MSH6. They are needed in order to carry out this repair function. I'm not going to go into the details of it, but what happens if this mismatch cannot be corrected? Have a look here, what is happening here. In patients who have lost the good copies of these proteins, which carry out this particular pathway to repair the mismatches, you can see there is a mismatch, AC, and this has been now corrected as a result of this. But what if we don't have this good copy of these enzymes carrying out this function, it can actually lead to lots of different diseases, including the this type of polyposis HNPCC cancer. And you can see from the endoscope, the cancer is quite obvious. If you take out these tissues and do histological analysis, you can see here this is a histology of this colon tissue, and you can see these are nice colon cells sitting here next to each other. These are all, of course, sadly cancer cells. But what is remarkable is that there is an infiltration of the lymphocytes. These are the cells which are trying to attack these tumors and kill them. That is exactly what Dr. Aziz was just mentioning, that these cells, which have these mutations, which they cannot correct, will lead to a lots of new antigens being formed, meaning new proteins which do not exist in normal cells. And as a result, they will invoke an immune attack, as you can see from this slide. What you can also do is that we were talking about this deamination, which is spontaneous. We have repair pathways in our body which can take care of this. It's called base excision repair, which can actually revert back any deaminated DNA into a wild type DNA. There is a whole process of it. As I said, I'm not going to go. This is at best MSc first semester stuff that you can study. And this is the last pathway, nucleotide excision repair, which becomes active when there are bulky adducts formed in the DNA. And the function of this is very simple. The nucleotide excision repair proteins would repair any bulky adducts which form, and as a result, they distort the structure of DNA. So when I was talking to you about the BPDE, what it was doing, caused by benzopyrene exposure, it is creating a bulky adduct. Well, these thymine dimers, again, I'm not going to go into the details, exactly doing the same thing. They're causing the bulky adducts to form, triggering this NER, or nucleotide excision repair system, as shown in this slide, taken from, I think this is from one of the textbooks. I gave you the link yesterday. So what happens if this system fails? You have illnesses like skin diseases, xeroderma pigmentosum, which can make these patients more prone to developing different types of skin cancers and not having these. So nucleotide excision repair is very important. It keeps our skin safe from harmful rays of sunlight, so that we don't develop illnesses, sadly, as shown on this slide. But nucleotide excision repair could be bad when we are treating cancer patients. Have a look at this review, and this shows exactly the same thing. So one of the chemotherapeutic drugs, which is used, I would say, mostly to treat a lot of different cancers and has been very successful, is cisplatin. And you can see here there are evidences, irrefutable evidences, that patients in which cisplatin initially works, but then it fails to work, it turns out that the nucleotide excision repair becomes activated. And do you want that repair system to be activated? No, you're trying to kill those cancer cells by damaging their DNA beyond repair. So activating an excision DNA repair pathway would be a disaster for that individual undergoing cisplatin therapy, because cisplatin will stop to function. So these are some of the points which I have summarized what we have discussed. I will try and now jump on to the tutorial today, which is harnessing the tumor defects for its own demise. Am I audible, everyone? Dr. Aziz, can you hear me? Perfect. Sorry. Okay, so let's crack on with this tutorial. So we discussed, I was showing you those migratory birds and so on, that these, uh, the cancer cells are very smart. They have undergone extensive training, but in doing so, they actually make themselves in some ways more vulnerable. Shall I say? In other words, they have some tumor defects. And because of these defects, if we can identify those defects, we can kill them. And you might have seen, if you visited London, these scaffolding sometimes in around the Big Ben and the Parliament in London, these scaffoldings as a part of restoration. Well, this scaffolding is not restricted to buildings and structures, but also to the DNA. So the previous lecture I was showing you just now, these nucleotide excision repair pathway, base excision repair, I touched very briefly, and mismatch excision repair. Many of these pathways actually need this molecule called PARP. So PARP is very important molecule, and its role is very well known in single strand break repair. You would say, hang on a minute, you are saying single strand breaks are taken care of in the body. It is double strand breaks we need to worry about. That's correct. But single strand breaks, I also said, if they are not really taken care of, they can convert into a double strand break. So PAR function is to make sure any single strand break is not converted into a double strand break. You can see what it does. At single strand breaks, PARP actually makes a polymer of itself. And in doing so, it actually creates a scaffolding to recruit other proteins, other DNA repair proteins, which are needed for the repair of DNA. So all these pathways I just showed you, these nucleotide excision repair, mismatch excision repair, the best base excision repair, all of them will have some role, and they will need part one to start this scaffolding. You need that scaffolding to recruit these proteins. So that is needed. Uh, I've shown this slide already. So this one already. So what happens here is that there are proteins which carry out repair of DNA double strand breaks, because we cannot really rely on PARP. It is one of the, if you see any immunohistochemistry, immunocytochemistry slides, you would see that one of the most frequently abundant protein in cell nucleus is none other than PARP. It is very rich, the cells in PARP. You can see what is happening here is that there are proteins which are there to repair the DNA double strand breaks. Where do you get double strand breaks? Well, radiation, treatment, cytotoxic treatments, platinum drugs, all lead to double strand break formation. We know that very well. But sometimes, if single strand breaks are there and they are not repaired, they can become double strand break in daughter cells. We know that as well. So what happens here is that if they are unrepaired, they will become double stranded. And this double strand having inside DNA two phosphodiester bonds broken in opposite strands will trigger a response called the double strand break repair response. And this response, it is coordinated by none other than BRCA protein, which I was telling you yesterday is a growth protein, but it is degraded, it is removed by the formaldehyde, which is produced. One of the things produces it is alcohol consumption. So what happens if you remove this? That the cells will not be able to carry out this entire pathway to repair the double strand break. So you need that pathway, BRCA pathway, to repair any double strand break, which is a product most of the time in cells when the single strand breaks cannot be repaired. So you have these other complexes like the single strand break repair pathways. I did not go into the details. I'm not going to go into the details of this pathway either, but I'm telling you more high level and giving you more high level information why this is important. And I hope you will be able to understand that. So what is happening here? Many of them will activate this kinase called ATM. ATM is a sensor of DNA double strand break. We all know that. And ATM is going to again amplify this DNA double strand break repair, in which the histones will be phosphorylated. We know that. And there are proteins called RAD will be recruited to the DNA, and this DNA will be repaired through homologous recombination. So the role of BRCA is very important in all of these pathways, the recruitment of these complexes, the ATM activation, and the recruitment of RAD51. BRCA is testing is done very regularly in many Western countries, even in Eastern countries, including India. And it has been noted that people with first degree relatives who died of cancer, such as breast cancers, who have BRCA mutations, their relatives, first degree relatives, will be much more susceptible to developing a BRCA related other cancers, because BRCA is a good gene that protects us. We discussed yesterday what degrades BRCA is alcohol can degrade BRCA. So cancers, many cancers will not have BRCA function, meaning if there are double strand breaks, they will not be able to uh, repair those double strand breaks because they need BRCA to repair double strand break. Does that make sense? So what happens is this is something which is dubbed as synthetic lethality. It's a complicated slide, but I'll try to make it easier. Imagine gene A is nothing but something that allows cells to take care of any single strand breaks. You have single strand breaks, and gene B is like gene BRCA, whose function is to repair any DNA double strand breaks. So you have single strand breaks, you cannot correct single strand breaks. Then you have gene BRCA or B, which can correct the conversion of any single strand breaks into double strand breaks. Both pathways are important here. But what happens if gene A function is gone as a result of mutation? Meaning, you have single strand breaks, they will be converted into double strand breaks. But double strand breaks will be taken care of because gene B of BRCA is intact. The cells will stay alive. This is what is happening in cancer cells. This is what is happening in normal cells. Normal cells have repair systems to repair single strand breaks, but they also have systems to repair double strand breaks. Cancer cells may not have the system to repair single strand breaks, allowing cancer cells to convert any single strand break into a double strand break. And double strand breaks, if they have it, BRCA may be there, and that can help. What happens in women with breast cancer is that BRCA function, as I mentioned on the last slide, the BRCA function is not there, but they might have single strand break repair system, which is still intact. As a result, cancer cells will still survive. Why? Because single strand breaks will not be converted into the replication cycle into a double strand break. If there is no double strand break, you don't need BRCA. So BRCA is a start because many women are not able to convert to repair their DNA double strand breaks. These cells essentially should have died, but they're not dying despite having double strand breaks. So they are already becoming fitter than normal cells who need to repair their double strand breaks. These cells don't. So what happens in synthetic lethality is, let's say BRCA gene function is off in many women, you can make inhibitor to block any single strand break being repaired. So if you block this, what will happen in cancer cells? That they will die. And that is exactly what is shown in this next slide. What you can see here is that there are proteins like PARP inhibitors. One of the examples will be olaparib, which is a clinical drug used across the world to treat many forms of ovarian, breast, and prostate cancers. Now, and you can see how does it function. If DNA has any single strand break, which I discussed with you, is happening all the time, we generate spontaneously single strand breaks in the DNA as a result of the repair process. Any of those pathways, mismatch repair, base excision repair, nucleotide excision repair. If you go and have a look, revisit those slides or your books, you will find there are always some part of the pathway which generates single strand breaks. And what is happening here? There are single strand breaks. PARP is there, forms a scaffolding, and then the cells will never have these double strand breaks. But what if you block PARP function? If you block PARP function, then these single strand breaks can be converted into double strand break. Okay, very good. What happens then? It turns out that PARP inhibitors don't really work on normal cells. They don't kill them. Why? Because normal cells will be able to have their double strand breaks repaired through the homologous recombination pathway, thanks to the BRCA which they have. So normal women, the breast cells, epithelial cells will have a BRCA. Even if you block their single strand break repair system, which will convert the DNA into a double strand break containing DNA that can be repaired by BRCA. So, what about women who don't have? Many of the women have germline loss of BRCA function. They don't have any BRCA, and as a result, what happens here that these women, when treated with inhibitors of PARP, their tumors will die. Their tumors, the cells will die because they are not able to repair the double strand break for which they needed BRCA, and that is absent. So you can actually have development of a therapy by which you can convert any single strand break, which are normally non-significant. They are generated in their millions in every cell all the time. You can make that into a double strand break. Normal cells will not be affected because they have a system, BRCA, thanks to BRCA, that they can repair these double strand breaks. The cells which give rise to cancer as a result of having BRCA deficiency now, because they don't have a good copy, because they're defective in repair of double strand breaks, you can make them more vulnerable to PARP inhibitors. And that is what is called synthetic lethality to treat breast cancer, women with breast cancer using drugs like olaparib, which are PARP inhibitors. Uh, so basically, you can imagine this situation is like this. PARP inhibitors normally don't do anything to normal cells who have an intact double strand break repair, homologous repair system in place. But in women who have don't have BRCA, what will happen? The cancer originated because there was no BRCA in the first place. So what will happen? Any single strand breaks which are being generated by normal cells and cancer cells alike all the time, those single strand breaks will become lethal as a double strand break. So you can imagine this as a barbecue causing a wildfire in a Californian desert, causing cancer to be treated. And this is an example of synthetic lethality in breast cancer. You can see there are some cells in breast. Not every epithelial cell is a cancer cell. You can see there are also normal cells, and normal cells have a good copy of BRCA repair pathway gene, and you can see here any single strand break, which is happening, as I said, all the time. PARP inhibitor will convert them when taken orally. It's a capsule, so every cell will be exposed to PARP inhibitor. Sorry. So what will happen here is that any single strand break will be converted into a double strand break. But normal cells have good copies of BRCA gene, and they will allow this double strand break to be corrected, and the normal breast cells will survive. What happens if the tumors if they are heterozygous for BRCA, meaning they have a good copy and a bad copy of BRCA? These cells will actually form resistant tumor cells because they are pretty much like these cells here, where the PARP inhibitor can convert single strand break into a double strand break. But because of the virtue that they have a good copy of BRCA, they're not going to die. They will become resistant to PARP inhibitor olaparib. What is going to happen over time? One of the things happen in cancer is the loss of heterozygosity. As a result, what will happen? Some breast cells shown here in this dark purple is that there will be loss of both copies of BRCA. So they don't have any system to repair any double strand break like this situation or like in this situation. And as a result, what will happen? You will have single strand breaks all the time. As I said, PARP inhibitor is going to convert it into a double strand break. In normal cells or in resistant cells, this will be corrected by BRCA. These cells have no good copy of BRCA, and as a result, these breast cells, which will be the most dangerous ones, having lost both BRCA copies, will eventually die. And you can see here, this therapy will only work if there are no good copies of BRCA. Even if normal cells who have a good copy of BRCA or both good copies, they will not be affected, meaning there will be less side effects of PARP-based therapies. Uh, so you can see here what we are doing with aldehydes, formaldehyde, alcohol consumption. We are decreasing the level of BRCA gene. So there may be good copies of the gene present, but you may not have sufficient protein itself. So there is no change at gene level, but there is less protein, meaning what will happen in the first place? These cells might actually become cancerous. And once they become cancerous, then you can see the chances are enormous for them to kill this patient. So, first of all, there are changes happening at germline as well as somatic mutations. On top, the environment is creating the deficiency of good genes, in this case by formaldehyde, which can lead to basically breast cancer. And it is known women who drink are more prone to developing uh, forms of breast cancer non-existent in non-drinkers. So what we did in the lab is, this is a paper we found that there is a similar relationship between the not BRCA and PAR, but PAR pathway and androgen receptor. I'll tell you in the next lecture what androgen receptor is, but have a look this double strand break repair pathway I was showing you before. Like BRCA in breast cancer, androgen receptor repairs DNA double strand breaks in prostate cancer cells. So, okay, so BRCA is not there. Yes, BRCA is needed, but androgen receptor, what we found, is needed to carry out this double strand break repair pathway. So what will happen? Nature is the smartest guru we have. So what happens if you block, and this is seen in breast cancer as well, if you block this androgen receptor, you see what happens here. This is the slide from a patient who were given therapies to block their androgen receptor pathway, which, by the way, is a regular standard of care treatment, block their androgen receptor signaling axis. And you can see this is the biopsy before androgen receptor blocking therapies and after androgen receptor blocking therapies. Can you see what is happening to the PARP activity? So PAR is the moiety which is added to the DNA by PARP enzyme. You can see here PARP enzyme becomes super active after we have blocked the androgen receptor signaling axis. So you have enormously high level of PARP activity. Normally, the PARP activity is very, very low. But when you block androgen receptor, the PARP activity goes up. What is the rationale for this? Well, that's a perfect rationale, because if system realizes, if nature sees that they don't have a DNA double strand break repair pathway, why not enhance single strand break repair? So if you can use PARP, you can enhance the PARP activation, you can probably inhibit the single strand break converting into double strand break in the first place. And that is exactly what happens in nature. When the cells realize they cannot afford to have any double strand break, they will upregulate the single strand break repair pathway so that there won't be any single strand breaks. And in the S phase of cell cycle, they will therefore not be converted into a double strand break. So what happens to treatments of tumors with inhibitors of PARP and inhibitors of androgen receptor? Have a look in this tumor xenograft model, and what it is showing you on y-axis is the tumor volume versus the time of incubation when the drug was given. You can see this is prostate cancer cells. They grow very well in mice. What happens with olaparib, which is a PARP inhibitor? Basically nothing. Why? It is not toxic to cells, because single strand breaks will be converted into a double strand break by olaparib-like drugs, which is a PARP inhibitor. It doesn't matter because androgen receptor is still active in them. So, no effect on tumors. What happens if you give androgen receptor inhibitor bicalutamide? Well, very little or no effect at all actually on tumors. This is not significant. Meaning, and if you block androgen receptor, it doesn't matter. I'm sorry about that. You can see what is happening here. If you block, sorry, the drug androgen receptor inhibitor, uh, what happens? There is no effect at all. Why? Because the single strand break will be repaired by PAR pathway as we saw here, but it is only when you block both of them. So you do have a single strand break repair pathway compromised in this group. Many of them will have double strand breaks, and these double strand breaks don't have the androgen receptor pathway intact. As a result, this will be converted into a double strand break, and those cells will then die. So this paper, we discovered this in over 15 years ago, published this in 2017. It was covered by print media. There were trials done on this, and based on this, this is a recent Nature Medicine paper, which actually shows what we showed in mice, that there is an effect, there is a benefit of blocking both PARP inhibitor, blocking PARP pathway and the androgen receptor pathway. Um, and the combination can lead to more tumor death. They use this treatment, combination treatment, and actually found that this is actually very effective in treating patients who have advanced prostate cancer, in which combination of this gives them a prolonged life. And they did thankfully quoted our paper, and they've received FDA approval of this therapy, which is now used throughout the world. I'm pleased to say for the treatment of advanced prostate cancer, combination of PARP inhibitor, olaparib, and other drugs which inhibit PARP, in addition to androgen receptor inhibitors such as abiraterone. So thank you very much. I'll stop with this and happy to take any questions for this tutorial. We have 14 minutes for any questions that you would like to ask me. So Swati, I'm not sure if you have activated the mics that they can ask me questions, the participants. Um, I'm more than happy for them to type their questions and I can try to answer. Although my voice, as you can see, is not very good these days, but I hope I'm understandable. Thank you. Thank you. Thank you. Dr. Aziz, any questions, any advice, any feedback, or from anyone else? Sure. Mm. So thank you, everyone. We will. Thank you, Swati. Let's reconvene tomorrow. Oh, there is a question. There is a question actually. So Happy is asking a question. Currently, my third year of BSc Biotech. So I did pursue a career in physician medicine. Although this course is primarily designed for master level higher, let's therefore utilize in the field. I would greatly value insight on how to undergraduate can strategically prepare. I think Hafi, what you need to do first of all. I was in a similar dilemma when I did my masters in Jamia Hamdard. I first and foremost tried, and during my master, I thank God I cleared CSIR, and they started back then ICMR. That was the first year. So I qualified those. I took admission in JNU. I qualified their exam as well. Uh, it would have taken. They had facilities, but I realized it would have taken 5 7 years at least to complete the PhD. That was not something I was hoping for. And there were issues, of course. I'm talking about 2003. Actually, yeah, 2001 actually. So what I would suggest is to do some substantial training, let's say 1 year training in a good research lab, and there are plenty of such labs in India. Make sure you're able to make some contribution to the research and publish research articles. So I understand most of you are super bright students and will graduate in your BSc or MSc with the first class. On top of this, what will attract future, you know, employers is your ability to demonstrate in your CV that you have actually meaningfully contributed to a publication. Have yourself as an author on the publication. This is what I've done. Believe me, I have second year BSc students in my lab. They 2 weeks ago finished writing a paper on which two of them appear as second authors, equal contribution. That is what I want students to do, but they time out 15 months from their conventional degree. So instead of 3 years, they have a 4 year degree. But I ensure that anyone who does that in my laboratory spend a lot of time, hard work, uh, pays off. And then as soon as they finish their BSc, they are on track to do PhD. Most students who I have in my laboratory now and previously never had any masters. They came right after their BSc and having spent a year in a top lab in UK or in Europe elsewhere. Sadly, for me to take any students from India is difficult because the tuition fee for Indian students is about 25 lakhs per year. Uh, for UK students, it's cheaper. It's about 4 or 5 lakhs. What would be best is I was in the same situation when I was looking for PhD studentships, so I had to write my own grant to support myself, which was thankfully successful, and I joined a laboratory with which in Germany, with which I wrote a grant application. And the guy, my supervisor, was honest enough to see my potential, invited me on that grant to do my PhD, paid stipend. My tuition fees was about 100 euros, which is 10,000 rupees or something, compared to in UK, which is 25 lakhs. So the quality of education you get in Germany is very good. Having said that, if you're able to get any positions in UK which are Wellcome Trust funded, any PhD studentships which are Wellcome funded or Cancer Research UK funded, usually they cover the tuition fees for international students as well. So I hope that answers your question. Any summer internships opportunities. Safi, I don't sadly take any students, even local students who have no visa problem. I don't take anyone because it creates to train students. It take a very long time, and I don't have a very big lab. I have four people working in my lab at the moment, and we don't have very much time that we can spend on training short-term students. So sadly, I have to disappoint a lot of students. I decline any short-term request. I take students if they want to come at least for 12 months, 15 months. And any of them who've done this usually will get a good PhD studentship elsewhere because they can produce evidence of having published paper when they're BSc students. So certainly a lot of employers, a lot of supervisors in different universities are interested in taking those bright students. And one of the criteria I have is that they should have very high first class marks so far in their degree, because otherwise I normally find it's a waste of their time and mine. Thank you. I hope so. Fatima is asking another question. She's pursuing chemical engineering from AMU, wish to pursue research in this field. Uh, what skills should I focus on and how can I academically pursue it? I think, Fatima, certainly if you're interested in this area, one of the things which is currently a hot topic is medicinal chemistry. And of course, you see this click chemistry being used in designing drugs and so on. So certainly if you chemistry, I have to accept research in chemistry. India is one of the at the forefront of chemistry research. So certainly if you're able to do your research, publish some papers while you're doing masters, which is quite a possibility. You can look for PhD studentships. I have to say, in Europe, there is no tuition fees as such. On top, you get your stipend, which is many times, depending on how spendthrift you are. Normally, it is sufficient to keep you get by during your PhD years. But you have to realize you're not on a job. You're getting a degree and then getting paid. I love that aspect. I do something cancer research, which is at the heart of my professional life, and I'm paid to do that job. So if you're able to publish some papers while you're doing chemical engineering degree there, certainly you would have much better chances, especially if it is in the area of medicinal chemistry. Of course, you know your field better. With I do collaborate with medicinal chemists, among others, and used to know someone who was one of the persons working here. He was a graduate of, he did his PhD from AMU. He was a top scientist, medicinal chemist in one of the top pharmas. I've never not met him in last 10 years, but definitely there are much more scope for chemical engineers in medicinal chemistry area. Swati, are there any more questions or? Perfect. Thank you. Thank you. Okay, we'll meet tomorrow then. Thank you. Take care, everyone. Thank you for organizing this, Swati and Dr. Aziz. Bye, everyone.